What is known about the content of our cells is that roughly 70% of them are made up of water. In 1989, scientists found that this number can vary greatly. For example, in some cells, it can be 55% and all the way up to 78%. So if this is the case, then how our organs, including muscles, maintain their structure? Well, your organs are made up of a large number of cells that have specialized function. For example, your heart, your liver, your kidney. So how do they withstand the harshness of everyday life? And how do they withstand the impacts they receive when we are playing sports? The answer to this question lies in what we find between the cells in our organs. That is, the structures that serve as scaffolding. This scaffolding is found in the space we call extracellular matrix. The extracellular matrix has many functions, and giving the structure and support to the organs is one of them functions. The extracellular matrix is made up of many substances, but consists mainly of protoglycans that are complex chain of protein core combined with glycosaminoglycans, and we will talk about these further in the video. And the second component are the fibrous proteins, and it's these two types of components that mainly give the organ the structure and the rigidness. In this video, let's talk about the protoglycans. There are roughly 43 genes that code for protoglycans. However, the actual number of protoglycans is much larger. So how is this possible? Well, it's through something we call alternative splicing. So what is alternative splicing? Our biology taught us that the genetic code that consists in the shape of DNA is converted to RNA before becoming actual proteins. And in this process, the regions of the DNA that code for the proteins are taken from the DNA and combined together. But when it comes to alternative splicing, this collection of codes can be different to one another, but may come from the same area of the genetic code, and therefore produce a different protein. But let's not go into that too much, and let's move back to our protoglycans. Now, the two most common types of protoglycans are chondroitin sulfate containing and dermatan sulfate containing protoglycans. What I mean by these words is that if you remember, I said earlier in this video that protoglycans are made up of protein core combined with glycosaminoglycans, and chondroitin sulfate and dermatan sulfate are glycosaminoglycans, or in simple terms, GAGs. GAGs are repeat units of disaccharides, or simply put, two sugars combined and repeated in a long chain. So it's the protoglycans and GAGs that take the hit throughout our life, and this is one of the reasons why they have to be recycled all the time. When the machinery involved in taking care of these GAGs go wrong, there comes the diseases that are mainly associated with deformities. One of the well-known group of diseases involving the recycling of GAGs is known as Mucopolysaccharidosis, or MPS for short. So let's see what happens in MPS. Well, the GAGs that usually get degraded by the cell's recycling center or lysosomes is unable to break these GAGs down, so they accumulate in the cell and outside the cell. But there are now some treatments that have been shown to be beneficial for people that suffer from these diseases that are associated with GAGs, or are scaffoldings in the organ. And this is where the people suffering from MPSs are given enzymes that break down the scaffolding through infusion to do the proper job and recycle these GAGs.